Hello, this is our lecture number 18, which is on research methods in behavioral neuroscience. And we're going to be taking a look at some of the major methods that are used, certainly not all of them. But I want to give you a little bit of background in just how behavioral neuroscientists use the in some cases, very sophisticated equipment and technology in order to do their research. I want to say at the outset that, again, this will not be an exhaustive examination by any means. What I will be doing is focusing in on some of the more frequently used techniques. And then as we go along in certain areas, as we advance along in the course, there will be certain very specialized kinds of methods that will come up that I will be talking about so that you can understand the research that is being done. I want to tell you a story, and it's one that you're going to be seeing a videotape on, and it's one of the required videos that I want you to watch, and it's on this very important story of Phineas Gage. There you see Phineas Gage holding in his hand what's called a tamping iron, and that tamping iron actually went through his brain, went through his cheek, right here, and then up through the frontal area of his brain. And the important thing is that he survived. And this is one of the most incredible stories really in the history of science, which has become so important for the discipline of psychology, and in particular the discipline, the specialty of behavioral neuroscience. So talk a little bit about this very interesting story. 1848 in Cavendish, Vermont, and again, you should have watched the videotape on this prior to this lecture. Phineas Gage is working on a railroad, laying tracks and blasting through rock in order to lay these tracks that are going from Boston, Massachusetts, up through western Massachusetts, then into Vermont, and then ultimately up to Canada. And he's a foreman. He's a leader of this group that's working on laying these tracks. And part of it involves blasting through, dynamiting through these very large rocks. And Gage is an expert in this area, and an accident occurs where this tamping iron that is used in order to place dynamite into these holes and rocks that have been created, all of a sudden it explodes, and that tamping iron goes through his brain. And this is a representation of where it actually went through, again, through his cheek, and then through the frontal area, and then out through the top of his brain. So again, the charge accidentally explodes, and the result is that Phineas Gage shows dramatic changes in terms of his behavior after. It's a shock that he survives, but before the accident, he was very serious, very hardworking, very energetic, very industrious individual. And after the accident, his behavior changes dramatically. Again, it's a medical miracle that he survives, but his behavior becomes very childish. He becomes very irresponsible, thoughtless. And indeed, he can no longer really function in the role that he was as the leader of this group that was laying these railroad tracks. This is so important because it really establishes the first documented evidence of the brain. If it's damaged, if it's not functioning properly, is going to cause changes in terms of behavior. So again, this is the first real documented evidence of this, and that's why it's such a pivotal point in the history of behavioral neuroscience. This is Cavendish, Vermont, that you see here, very small village in central Vermont. This is the actual area where this happened. These are the tracks that were laid, and they were blasting through these rocks in order to create this track going through this area. This is an artist rendition, a cartoon actually, that was in a local newspaper in Vermont at that time. But this is something, a news spread of this all over the world, really. And this, if you go to Cavendish, Vermont, this is a plaque that you see on the village green, which summarizes the events that occurred, the chronology of events that occurred during this very interesting situation that emerged. And this is an actual write-up from a newspaper in Ludlow, Vermont, which is right near Cavendish. And it says, as Phineas Gage, a foreman on the railroad in Cavendish, was yesterday engaged in tamping for a blast, the powder exploded, carrying an iron instrument through his head, 1 1/4 inch in circumference and 3 feet and 8 inches in length, which he was using at the time. The iron entered on the side of his face, shattering the upper jaw and passing back of the left eye and out at the top of the head. The most singular circumstance connected with this melancholy affair is that he was alive at 2 o'clock this afternoon and in full possession of his reason and free from pain. So again, this travels, worldwide. This incredible accident that occurs in 1848, picture that you see here is the actual skull of Phineas Gage. This is at Harvard University in what's called the Warren Anatomical Museum. Here's Gage's actual skull, and here is the tamping iron that went through his brain. It's very interesting. Phineas Gage survived, I believe, another 12 years or 15 years after this. He became a much sought after individual, for example, for traveling circuses, that people would pay money to see him and to meet him and to see the tamping iron that actually went through his brain. This is an incident that obviously had an incredible importance for the history of behavioral neuroscience and for science in general, because now many believe that we had to know more about the brain. And we have to know more about the anatomy of the brain. We have to know more about the brain in terms of its relationship to behavior. So again, this is a pivotal point in the history of science. If you take a look at a little basic anatomy here, here's our brain. Here's the frontal cortex of our brain right here. Here's parts of what we call the old brain. Okay, the frontal cortex is considered to be new brain, and new because it's a more recent adaptation, evolutionary adaptation. But here's our old brain here, which consists of the thalamus, the hypothalamus, the hippocampus that you see right here, the amygdala, and also the olfactory bulb. That tamping iron went right up through here. Okay, right up through here. So what happened was that the pathways, the neural pathways that ordinarily connect the smart part of our brain to the older, more primitive part of our brain that's involved in the regulation of emotions, for example, basic motivational processes. Now that had been cut off, and the smart part of our brain was no longer in a position to be able to suppress what was happening with the more primitive part of the brain. So indeed, this is what happens in the case of Phineas Gage. He becomes more emotional and less thoughtful in terms of his behavior. So that in and of itself was important and helped us really to understand how these two different parts of the brain might be involved in our behavior. The new brain, the frontal cortical area, the smart part, involved in higher cognitive processing, thinking, reasoning, decision making. The old part of our brain here involved in more basic kinds of motivational processes. So that in and of itself was a very interesting finding that helped us to understand, a little bit more about the functioning of the brain. So these research methods, we're going to be taking a look at a number of different ones. And again, I'm going to be focusing on just a few of the older ones, more popular ones, that are more recent ones that are going to come up that we'll focus on when we get to, individual kinds of research questions and individual areas of research on different behaviors, for example. But we're interested, these techniques involve visualizing the human brain, being able to do it accurately, recording psychophysiological activity. In some cases, we're using methods that are invasive in order to actually manipulate or change parts of the brain. There's pharmacological research methods in which we are exposing the brain to different types of drugs. Neuropsychological techniques that we use in order to assess level of brain damage, for example. Behavioral methods of cognitive neuroscience in which we're assessing decision making, for example, and how that is related to different changes that are occurring within the brain. There are also methods, paradigms that are used to study animal behavior and to very specifically, manipulate the brain, maybe biochemically, maybe in terms of removing parts of the brain. And then there's the whole area of genetic engineering, which is going to crop up in a number of different areas that we're going to be exploring that have become truly just so advanced and interesting, and have helped us so much to answer various problems in what our behavior is a product of in terms of underlying biological basis. So let's begin this exploration. And certainly ways of assessing the healthy brain. This is a cerebral angiogram that you see here. Here's the major artery of the brain. And that, a dye is infused in the cerebral artery, and we can visualize the circulatory system, and we do it by way of an X-ray photography. And of course, this is very important in terms of understanding what may be happening in terms of the rupturing of an artery and the obvious implications of stroke. So again, being able to visualize the brain becomes very important, and it's an important tool in the field of behavioral neuroscience. CT scans, again, one of the older techniques that was used, still used today, but not anywhere near as much as it once was. A person, their head is placed inside of this device that you see here, and there's a very large magnet that rotates around the individual's head, and indeed by way of X-rays that go through the brain. We can then detect on the other side here, this by way of this X-ray detector, we can take pictures of the brain. And here you see a series of horizontal scans that have been taken. Indeed, the person here that's monitoring this, they get this, three-dimensional reconstruction that occurs in terms of, what the brain actually looks like. So indeed, this is one of the older techniques that's been used, called computed tomography or CT scans, popular way in order to explore the brain and various changes that may have occurred in terms of structural changes that may have occurred in the brain. This is called a color enhanced mid-sagittal magnetic resonance scan. So now these, unlike the CT scan, these are very, very high resolution images. And the way in which this is done is radio frequency waves in a magnetic field activate various hydrogen atoms. And that is what is allowing these very clear brain images, much clearer than what we would ever see in the case of a CT scan. So now this was the next level, really, of our ability to be able to visualize the human brain in terms of these very high resolution images. This is a structural MRI, and take a look at this. This is in the yellow, these are the ventricles of the brain. Again, this is a ventral view of the brain, and or dorsal view rather of the brain. And here is a tumor right here and right here that has been treated by way of radiotherapy. So here's what the MRI scan looks like. This is shortly after the treatment. And several weeks later, we can see that the tumor has actually gotten larger. Again, you can see very significant growth that is occurring here. So again, this is structural MRIs, very important as a diagnostic technique in order to explore the structure of the brain. In this case, we're taking a look at what is happening in terms of this tumor and how the tumor is infiltrating and growing. And we can do that by way of the structural MRI. How are these, how are functional MRIs actually done? Here's an example of how it's done. Here's an individual that's actually in an MRI machine, and we're going to ask that individual to perform certain tasks that might be some as simple as simply tapping their thumb against their fingers. Others in which maybe they look at a picture on a computer screen. But one of the things that we know is that when a patient is performing a particular task and then they're in that MRI machine doing that task, there's increased blood flow that occurs to the part of the brain that is most involved in that task. So again, this is another level really of trying to understand, relationship between our biology and our behavior. Here's a functional MRI that you see right here. These are parts of the cortex, and you can see in the red that when this subject was looking at strings of letters, and they were asked to specify which strings were words, you get this very profound activation that is occurring, and that's in the red that you see. This is in the right hemisphere, the lateral surface. This is also lateral surface, left hemisphere. And this is left hemisphere medial surface, and also medial surface of the right hemisphere. You're getting this profound activation that is occurring in those individuals. So again, this is fMRIs that are helping us to understand what is happening in terms of the surface activity of the brain when the individual is involved in a particular task. Positron emission tomography, PET scans, another very important technique that is used. It's very simple, okay, how it's used. A radio-labeled substance, which is called 2-DG, 2-deoxyglucose. Radio-labeled glucose is what it is injected into the carotid artery. And glucose, as you know, is a fundamental fuel for the brain. And it's rapidly taken up by neurons in the brain that are active. So if you take a look then at these horizontal sections in which we're taking a look at psychological activity, it shows specific areas of the brain that are active. So for example, when an individual is looking at a visual stimulus, for example, let's look at that one. That's on the left that you see right here. And again, here's the frontal part of the brain. Here's the back of the brain. Back of the brain we call the occipital cortex. When you see a lot of red lighting up, that means there's a tremendous amount of activity, reds and yellows, but the most activity is in terms of reds. What you see is when that individual is shown that a visual stimulus, you get this profound change that occurs in the visual cortex of the brain. So that's what that is illustrating. And again, take a look at these other ones. Here's when there's an auditory stimulus. Here's when there's a cognitive stimulus where a person is involved in decision making. Here you can see the frontal area of the brain absolutely lighting up. When a person is processing a memory, again, here you see a lot of lighting up that's occurring in these areas that you see right here. So again, this is another popular way, these PET scans, of taking a look at relationships between our biology. In this case, how much metabolism is taking place of glucose in various parts of the brain in response to a specific type of stimulus that the individual is confronted with. So again, another important and popular technique. This is called the paired image subtraction technique. Take a look at what is happening here. When an individual is shown a visual stimulus, again, we're in this case, it's a flickering checkerboard. And there's a lot of brain activity that is occurring. Here's the control condition that you see right here. If we subtract that control condition, and this is when the flickering checkerboard is not occurring, subtract that from when the visual stimulus is presented, you see this. This is the difference. And again, this shows the parts of the brain that are most affected and the parts of the brain that are most active. And it is clearly in the occipital cortical, the visual cortex of the brain. So again, this is another very popular technique, this paired image subtraction technique that is used. Here's a little bit of the basics in terms of the EEG machine, which again is an older piece of technology, still used certainly today for certain types of research in the field of behavioral neuroscience. Here you see a person with these scalp electrodes. Each one of those electrodes feeds into these this bank of amplifiers that you see here, which are going to increase that signal that is coming from that part of the brain, from those scalp electrodes. So each electrode now corresponds to an ink pen on this EEG machine. Again, those pens are connected to these amplifiers that are increasing the signal, electrical signal that's coming from the brain. And you have this paper. Ink is making a mark on this paper in response to those changes in electrical activity of the brain. They are showing these deflections that occur, which indicate that there's a current change that's occurring in that part of the brain. It makes a mark on the paper that you see here, and that paper is continuously advancing. So if you take a look at what the output looks like, here's a series of 8 electrodes that have been placed in various parts of the brain, and these are the pen deflections. We call them EEG recordings that you see, again, from 8 different scalp electrodes. Take a look at typical EEG. There's 2 fundamental parts of electrical activity of the brain that you need to be aware of. First of all, there's the amplitude of the wave. What we mean by that is how far from its base does the brain wave deflect. So here's the brain wave of a person who's aroused that we see right here. And you can see that there's a lot of deflections, meaning this is the frequency, the number of deflections that occur per unit of time, but they don't deflect very far away from the base. Contrast that with a person, for example, that's in deep sleep, and you don't see many deflections that occur. So this is a low amplitude brainwave. Excuse me, a high amplitude brainwave, and the frequency, you don't see, very frequent departures from that hypothetical base that you see here. That is, they're deflecting very far away from the base, but you don't see very many deflections per unit of time. So this is a high amplitude, low frequency brainwave, which is typical of a person who's in deep sleep. And again, this high frequency, low amplitude brainwave of a person who is aroused. So again, those are the basic parameters of brainwave activity in terms of amplitude and in terms of frequency. What you see here are, four, methods really for recording electrical activity of the nervous system. Let's start out with this one first. This is called an intracellular unit recording, and I think this is one that you're already familiar with. This is recording membrane potentials from one neuron when it fires, and again here you see this firing that occurs, and again happening over a relatively short period of time. This is an extracellular recording. We're recording disturbances that are created in adjacent neurons when they fire. Again, this shows a firing right here and here and here and here and here and here. This is called a multiple unit recording that we see here, in which, that electrode is large and it's really recording from a number of different neurons, their action potentials, and we add them up and we plot them, and this is what it looks like what you see here, okay? And this is an invasive recording in which we actually implant an electrode into the brain, and we measure the EEG signals, and again, that's really not related to neural firing in any specific kind of way, and here's what that looks like. So these are four basic ways, really, of recording electrical activity from the nervous system. One of the things scientists do is they engage in this procedure that's called averaging of electrical brain waves in response to a very specific kind of stimulus. So, for example, if we were taking a look at what was happening in the auditory cortex of the brain, when there's no auditory stimulus, again, if you took these 1-second segments, it would look something like this, and then if we averaged them, it would look like this. Okay, not a straight line, but certainly, a relatively steady line. If, on the other hand, we were to present some kind of an auditory stimulus, a sound, and again, take a look at our 1-second segments. Here's where, again, we're sounding like a clicking noise. And again, here's these 1-second segments that you see here. If we were to average them, it would look like this, okay? So this is what it would look like in response to an actual sound, and we call these evoked potentials. We sound, in this case, it's an auditory stimulus, a clicking noise, and you get these very pronounced changes that occur in terms of electrical activity. Contrast that with what we see here, okay, where it's relatively steady. Take a look. This is very interesting. Portrayal that you see here. We're going to take a look at what's called P300 waves. Let's take that clicking noise and let's associate it with a rat, let's say, that's in a box that it's pressing a bar in order to get food. And shortly before the animal presses the bar to get food, or actually at the same time it presses the bar to get food, this click sounds. So the animal now comes to associate that clicking noise with the delivery of the reward, the delivery of the food. And if we take a look at what happens here, this is called a P300 wave. Now if we just sound that meaningful click, you get these very pronounced changes that occur in terms of the electrical activity of the auditory area of the brain. So this is called a P300 wave, and it occurs only if that stimulus has meaning. And in this case, that click, which is signaling that the delivery of food is going to come soon, that's a meaningful event. That's a meaningful stimulus. And indeed, it's being reflected in these very profound changes that occur in terms of electrical activity of the brain. this is an interesting illustration that you see here. What we're able to do now with EEG and MRI is really to combine them. And what you see plotted here is the distribution of theta waves. These are theta waves that you see here that are recorded while a subject is working on a memory task. And we can record not only those EEG signals, but we can record structural cerebral MRIs. So what you see then is those theta waves right there are occurring in the anterior cingulate cortical part of the brain. That's where we see a lot of this MRI activity, and it's associated with this very pronounced theta wave activity. So this is what happens when we combine the two. We can take a look at the two simultaneously. This is called transcranial magnetic stimulation, or TMS, a relatively new technique. We're going to talk about it a little bit when we get to the area of treatment of certain disorders like depression, for example. But this is a non-invasive technique in which we are, passing this wire coil, this magnetic coil, near the cortex of the brain, the dorsal area of the brain. And what it does is it passes these electrical pulses toward the brain, and it's altering action potentials. So we can use this TMS then in combination with fMRI and with PET scans as a way of assessing, what may happen in terms of various behavioral tasks that the individual is engaged in. And again, it's being used as a therapeutic tool in the treatment of depression. Here's a procedure, a piece of equipment that's very common in behavioral neuroscience. This is called a stereotaxic instrument, and there's a videotape that you'll be watching or should have watched on this. This stereotaxic instrument is used in order to fix a rat's brain in place. Anesthetized rat is placed in this apparatus. These ear bars that you see here go into the ear canal, the auditory canal of the individual rat. The rat rests its mouth on this device that you see right here. The ear bars are affixed to the skull. And that allows us to keep the animal's brain in one place so that it doesn't move around, that its skull doesn't move around at all. Scientists have developed what they call a brain atlas for almost every single mammalian species. This is part of that brain atlas that you see here, where we're able to actually map where different parts of the brain are located. So let's say that we wanted to put an electrode into the amygdala of a rat. Here's our amygdala that you see right here. And we know precisely how to place that electrode into the brain while the animal is in this stereotaxic instrument. So what we're going to do is we're going to drill a hole in the animal's skull. And by virtue of this map that we have, we know very precisely where the amygdala is. And what we're going to do is we're going to pass this electrode down into the brain. And the way in which we do it is by way of what we call coordinates. If you take a look at this right here, this shows the coordinates for the amygdala. That target site that you see there is 2.8 millimeters posterior to what we call bregma. And I'll show you that in another slide in just a few moments. 4.5 millimeters lateral to bregma and 8.5 millimeters ventral to bregma. So we have our animal now that's been anesthetized in this device. Here's bregma that you see right here. These are actual sutures that you see naturally occurring in the skull of the rat. And we know from this atlas that we have here, this map of the brain, that the amygdala, once again, is 2 millimeters posterior to bregma, 4 millimeters lateral, and 8.5 millimeters ventral. So all we do now is drill that hole in the brain, and then we lower our electrode down into the brain, and we can use that electrode to very precisely damage that part of the brain. Here's our animal now that has had this electrode placed into its brain. The animal is freely moving around. That electrode is fixed in place with acrylic cement. And it's allowed to harden, and what we can do is we can, for example, in this case, we can periodically stimulate the animal's brain and take a look at its resulting behavior. So again, this is an important tactic, an important tool that is used. A little bit more about this thing called bregma. Let's take a look at this slide that you see right here. This is what the human skull looks like. These are what are called sutures. These are little gaps that you see here between various bony parts of the brain. Here is what's called the coronal suture. It occurs in the same place in the skull of all human beings. This is what's called the sagittal suture here. And again, it crosses or meets this coronal suture at this spot that's called bregma. So again, that brain atlas that I showed you, the one for the rat, and again, let's take a look at the rat right now. Here's the skull of a rat. Here is that point called bregma, right? Here's our coronal suture that you see here. Here's our sagittal suture. And again, this is always occurring in the same place. So again, we can just, from this point, okay, we can always, we know precisely where to place those electrodes. And again, this is a popular tool that's used in the field of behavioral neuroscience to, for example, to stimulate that part of the brain and look at what is happening in terms of behavior, or we actually burn out small parts of the brain electrolytically. Or we place drugs down into the brain. Again, it's a tactic that is used very frequently in the field of behavioral neuroscience, and we'll be talking about it as we go along. Here's another popular piece of laboratory technique that's used. Again, here's our stereotaxic instrument. And this time what we're going to do is we're going to drop this knife, this subcortical device, down into the brain. And again, this is while the animal is anesthetized and while it is in this apparatus called the stereotaxic apparatus, and this little blade swings out and we can cut. In this case, we're going to cut a pathway. And we can explore later on, after the animal has recovered, we can explore what happens to the animal's behavior when that pathway has been cut. This is called a cryoprobe. Same kind of procedure. We're using a stereotaxic instrument to implant this down into the brain. And indeed, what we can do is we can either selectively cool or heat the brain by way of this cryoprobe that you see here. And again, these cryoprobes are very, very small, very, very tiny, and they don't cause very substantial damage at all to the brain. But we can place them into precise areas of the brain and look at what happens when the brain is either heated or cooled. This last slide is I just kind of threw in there because I think, you know, this helps us understand how sophisticated we've really become in the field of neuroscience. You recall we talked about Golgi stains. Here's a Golgi stain that you see right here. This is one of the first stains that was used in order to visualize neurons within the brain, their cell bodies and their axonal and dendritic projections. This stain that you see here on the right, this is called a Brainbow stain. And that Brainbow stain is, state of the art now. What we can do is we have labeling techniques in which we use different dyes, in which we can label individual nerve cells in different colors. And we can now track those axons and those dendrites over long distances in the central nervous system. I mean, just take a look at the incredible detail that this allows, and take a look at, the Golgi stain, which is very primitive in terms of its detail. So again, that, some of the basics in terms of behavioral neuroscience techniques, and we'll be taking a look at more as we go along, but these are some of the fundamental ones. So in our next lecture, we're going to be talking more about the cerebral cortex of the brain, a little bit more about the structure of the brain.